A-Level Biology 9700, paper 4, variant 2, May, June 2024. And question number 5 is the genes BRCA1 is expressed in breast tissues and in several other tissues of the body. BRCA1 codes for a tumor's breast protein. This protein is involved in either repairing damaged DNA or in triggering the death of a cell if DNA cannot be repaired. Some mutations in BRCA1 are associated with an increased risk of developing breast cancer. State one reason why a doctor would recommend that a person has a genetic testing for a mutation in the gene BRCA1 and explain why this mutation would put the person at a greater risk of breast cancer. Students, you know that everyone has two types of breast cancers genes in every cell of their body. When functioning properly, BRCA1 and BRCA2, they repair DNA. They keep other genes healthy and prevent cancerous changes in the body. But due to some mutations, they uh, they are damaged. Either of these genes or the person's risk of cancer is increases, because in this way their functional loss leaves some cells highly vulnerable to DNA damage, including damage that triggers cancer. So in this way, this is the reason that the person at a greater risk of breast cancer when the mutation occur. So this is all about this question. Now I would like the right answer. That is in this way that the female, you can say, in some of the families there is history of breast or ovarian cancers. This is also the female suffer with this problem or the disease. So we will start from this point, that is the some of the females have family history of breast or ovarian cancers. So those female are at high risk, you know, and if mutation occurs, these proteins in gene BRCA1, the structure of the protein is changed. Due to which DNA is not repaired. So increased number of mutations. So the uncontrollable cell division can lead to breast or ovarian cancer. So the risk is increased. Next question. Part B. DNA sequencing is one method used to test for mutations in BRCA1 before DNA sequencing occurs to DNA samples goes through the polymerase chain reactions or PCR. So suggest why PCR is used for testing for mutations in BRCA1 because only small amount of DNA is available. And, do, and the second is to amplify DNA. Next question, genetic screening shows that in the UK, 1 in 400 female have a mutant alleles of BRCA1. 70% of the females with a mutated allele of BRCA1 will develop breast cancer by the time they are 80 years old. So in 2022, the population of females in UK was 3.5 into 10 raised to power 7. So calculate the number of females in the UK population in 2022 who it is estimated will develop breast cancer by the age of 80 years old. Give your answer in the standard forms. Show you're working. So here you can see there are out of 400 females, every one female is have a muted allele of BRCA1. So we have to calculate in 3.5 into 10 raised to power 7 females. So in this we will write in this way 1 over 400 multiplied by 3.5 into 10 raised to power 7. So after calculating, the answer is 8.75 into 10 raised to power 4. Now we are moving. One advantage of genetic engineering is to determine which drugs should be selected to treat cancer. A study was carried out on women with breast cancer who has treated with a DNA damaging drug that kills tumor cells. The women uh, had all been tested for BRCA CA1 mutations. One group had BRCA1 mutation and the other group did not have BRCA1 mutation. The researchers used data to assess the probability of survival for the women in each group for the study of treatment with the drug. So you can see the graph, and graph shows the data in which the probability of survival for a time period of up to 180 months. 180 months, and you can see the probability of survival are greater in group with BRCA1 mutation, and the other group which with no BRCA1 mutation, they have probability of survival, low probability of survival as comparison to other group. So the researchers suggested the hypothesis that the DNA damaging drug is more effective in women with BRCA1 mutation. Discussed whether the results in figure 5.1 support the hypothesis suggested by the researchers. So this hypothesis supports that the with women with BRCA1 have a higher probability of survival as comparison to the other smaller rate have there is a smaller rate of decrease ability of survival. So next question number 6. And question number 6 is a diagram of part of a mitochondrion, the cytoplasm, in the mitochondrial membrane and mitochondrial matrix, outer membrane, inner membranes, and in the inner mitochondrial membrane, A, B, C, D, different types of proteins has been shown. And you can see the question is figure 6.1, use arrows to show the route taken by oxygen from the cytoplasm into the mitochondrial matrix. So oxygen will enter into the mitochondrion from the cytoplasm. So it will diffuse through this membrane from the cytoplasm into the inner mitochondrial inner membrane space, and from the inner membrane space it will diffuse into the mitochondrial matrix. So we will draw the arrows in this way. Now the next question is identify what is D. Identify the D and the group of proteins represented A, B, C, and describe their role in oxidative phosphorylation. So oxidative phosphorylation is actually you can see that this is D is the is the enzyme ATP synthase, and the other A, B, and C are the collection of proteins which are bound to the inner mitochondrial membrane and organic molecules, and which electrons pass through in a series of redox reactions and release energy. So the energy released forms a proton gradients, which is used in chemiosmosis to make a large amount of ATP by the protein ATP synthesis. So in this way, the oxidative phosphorylation takes place. So I would like all these points in the way that is the D is oxidative ATP synthesis, and A, B, and C are the proteins inner membrane which transport the electrons. Energy, and during the transportation along electron transport chain, energy is released. So the transportation hydrogen ion, or we also call them as protons, into intermembrane space So concentration of these hydrogen ions will increase there. So they will move, diffuse, from intermembrane space to matrix. Then synthesis of ATP from ADP will take place. Next question. The matrix of a mitochondrion contains small circular DNA. In the cells of the humans, the mitochondria have circular DNA that contain 37 genes. So suggest functions for these mitochondrial genes. So you can say the mitochondrial genome contains 37 genes, and out of these 37 genes will encode 13 proteins, 22 transfer RNAs, and 2 ribosomal RNAs. The 13 mitochondrial genes encoded protein all instruct cells to produce protein subunits of the enzyme complexes of the oxidative phosphorylation system, which enables mitochondria to act as the powerhouse of our cell. So we are going to write the answer now. So the mitochondrial genes are helpful, or they provide the energy for Krebs cycle or link reaction. Inner membrane proteins. So they move through channel or carrier proteins. And then the formation, you can see also the formation of transfer RNA, 22 transfer RNA and 2 ribosomal RNA formation will take place. So it provides the instructions to the cell to produce protein subunits of the enzymes complexes. So it was all about question number 5 and 6. Please like, share, and subscribe my channel.